Catalase test. Catalase enzyme is present in all living organisms exposed to oxygen. They produce hydrogen peroxide and in some cases extremely toxic superoxide. So that superoxide accumulation will result in the death of the organisms unless they can be enzymatically degraded. So organisms capable of producing catalase rapidly degrade hydrogen peroxide to oxygen and water. So the Catalase test is used to determine the microorganisms to degrade hydrogen peroxide into water and oxygen. So the materials required for the test is inoculation loop, overnight culture, Bunsen burner, hydrogen peroxide, H2O2, water, and the capillary tubes. So all the things we will arranged in the LAF, that is laminar air flow. So there we will work on our experiment. Now, when we start the method, hydrogen peroxide, this can be added directly to the culture or it can be done after adding the emulsion, making the emulsion. So firstly, the procedure for the slide culture. This Catalase test can be done by the two methods. First is the slide method, second is the SLANT method. So in the slide method, we can divide the slide into two sections with the marker pen. One should be labeled as test and other should be labeled as control. Now we will put a small drop of water on the test area and one drop on the control area. Then we will take the overnight culture and with the inoculation loop and the aseptic condition, we will take a small colony of the organism from the test tube. And again we will close the tube under flame. Then we will take this colony, transfer the colony on the marked field of the glass slide, emulsify the colony on the each drop to make the smooth suspension. Once the suspension is formed, we will take the hydrogen peroxide with the pipette and we will put the hydrogen peroxide over the test smear. Do not put anything on the control because that is a control. Now, after this, we will observe the result. As a result, the slide will be having the bubbles. So that will be the indication of positive result. So bubbles will form on the glass slide. That is a positive result. And if no bubbles are there, that will be catalase negative organism. And if the bubbles are there, that is the catalase positive. Now the same way we will go for the SLANT method. In the SLANT method also, we will take the hydrogen peroxide, open the mouth of the tube and we will put the hydrogen peroxide to the slant and that will show the bubble formation.